Hi, thank you for the introduction. Hi, my name is Young-Sun Kim from the Samsung Biologics MSAT team. The title of my talk today is Application of QbD, the Quality by Design approach for tech transfer, and then process characterization of the biologics. During my presentation, I will showcase how the QbD principle can be applied to the process tech transfer, the process characterization, as well as the process development. As a model, I can use the monoclonal antibody as a modality for the example of my presentation. Here's an agenda for today's presentation. After some introduction to my company, Samsung Biologics, I will gradually become more specific toward the main topics, including the QbD principles and then process characterization study. I'm going to cover the tech transfer, the process validation, and then PC study connected with the QbD application. At the end of presentation, I will show some case study with the monoclonal antibody case. Okay, let me start with a brief introduction of the Samsung Biologics, the business model at the high level. Ever since the establishment in 2011, Samsung Biologics has been providing the end-to-end services to our customers. While our CDO services support the process development of the client's candidate molecule from the DNA sequence to IND filing support, CMO services enable sustainable product supply for the clinical study as well as a commercial release. Bio R&D Center closely work together with the other functional centers to provide excellence in services and expanding our capability into different modalities such as the ADC and messenger RNA. Again, the Samsung Biologics is becoming an integral service provider supporting the expedited introduction of a client's molecule to the market across the different modalities. So we continue to expand our global partnership with the big pharma as well as biotech companies globally. Here is some data showing on increasing the market size trend for the North Americas, the biopharmaceuticals. As seen in this slide there, market sizes keep increasing until 2023. Even though the global graph is not shown in this slide, the market size also follows a similar trend to the North America. In 2032, the global market size of the biopharmaceutical is expected to be about $1,200 million. With the market size increase, biopharmaceutical manufacturing demand is also projected to increase consistently throughout the years. As Tim briefly mentioned during his introduction, Samsung Biologics has a capacity of 604 kiloliters for now. So far, the Samsung Biologics has a full plant at the Songdo, located in South Korea. We started with the 30K facility with the over 5,000 liter bioreactors. The Samsung Biologics kept expanding its capacity with the Plant 2 and Plant 3 and Plant 4. In the BioCampus 1, including the Plants 1, 2, 3, 4, Samsung Biologics has a total capacity of 604 kiloliters. Along with the DS manufacturing facility, the Plant 1 and Plant 2 have a DP production capability. The Plant 3 has a feature of N-1 perfusion for high cell density inoculation process with a 2 separate downstream suite. Plant 4A is a 15K facility, while Plant 4B has a 10K liter bioreactors. We are expecting to complete construction of Plant 5 by early next year, with the completion of the first phase of BioCampus 2 next year. The Samsung Biologics total manufacturing capacity will be 784 kiloliters. The new plant will have a fully digitalized facility with a simplified and standardized operation. We leveraged the lessons learned from the previous BioCampus 1 for this BioCampus 2. As mentioned earlier, Samsung Biologics is expanding our capacity. We'll have 784 kiloliters capacity next year, early next year. We also provide agility to streamline the process, our process, and optimize the timeline to accelerate the client's program, regulatory clinical program. Samsung also provide a flexible facility design, equipment, and then tailored service to meet the client's diverse portfolios. Samsung Biologics has a very strong track record to optimize the operation, especially the optimal operation to support a diverse portfolios of clients. The last one is technical innovation in order to provide improve the productivity as well as the process robustness. With these 5 pillars, Samsung Biologics is providing the very good operational excellence. With these operations, we achieved more than 98% the batch success rate, along with the 335 the regulatory approval from the regulatory bodies such as FDA and the EMA. Today's presentation is more focused on technical innovations using the QbD process to maintain our process more robustly to produce a consistent product yield. Okay, let's change gears and then start discussion on the production, the drug discovery and approval process. Typically, the getting the drug approved by the regulatory authority takes a very long time. It's a very long journey with a low probability. Often, a lot of lead molecules are dropped out during the preclinical or clinical stages for many reasons, such as the safety, efficacy, or the stability issues. For approval of biologics and process validation is a very critical step to go through for the BLA filing in the U.S. or the MAA filing in Europe to show the process robustness to manufacture the biopharmaceuticals. Here is some background information on the product life cycle of drugs and then the key activities for the commercialization of drugs. So you start with the target product profile shown on the left side. This target product profile is outline the desired profile of and the characteristic of the target product that is aimed at the particular disease or diseases. This one includes some desirable attributes of the product, from a drug product, including the safety, as well as the efficacy related to the characteristics. Such profile can guide the product research and develop for the drugs. Once this one is defined, with the product research and development, we can find out the QTPP, which is quality target product profile. The QTTP is the summary of the characteristic that the drug product should ideally have to ensure its quality and safety and efficacy. This is the first step in the developing the product, drug product using the quality by design principle. So once this quality, the target product profile is available, and we can do the product design, process development, and the process design. These steps are the very critical steps in the process to bring in to the new bringing the new product into the market. Biopharmaceutical process design involves development and implementation manufacturing process for the biopharmaceuticals. Once the process is locked in the middle of the process, usually tech transfer for the process to scale up is happening in the CMO. After confirming that the compatible product is manufactured at the larger scale, we go through the process validation, which is the late stage of this the product life cycle. Once we prove that this process is showing the robustness with the compatible the product quality, this product will be approved by the regulatory bodies. Once this product is approved, we're going into the commercial scale production. One thing that I want to emphasize in this slide is on the top of this slide, we have the control strategy in the green, the diamond. As you can see, the control strategy is all over the place, from the process development stage to the commercial stage. That means this process control stage can be evolved with a more understanding of the process during the process characterization as well as process validation. I mentioned that one of the critical steps for the product approval is process validation, which can demonstrate that the bioprocess is very robust enough to manufacture the compatible product between the batches. As you can see here, the process validation includes 3 phases. The stage 1 is a process design, including the process development and then process characterization. The stage 2 has a scale the PPQ runs in the GMP facility, and then the facility-dependent study like a buffer mixing study. Mainly the phase 2 demonstrate the consistent operation at the GMP facility. This one shows to the this process shows the compatible product quality between the PPQ batches. Stage 3 is after the product approval. This is called the continuous process verification. Once your product is approved, we are going through the commercial stage. During this commercial stage, we keep monitoring the process parameters, including the process variables, as well independent variable and then dependent variable, and then also we are using the statistical process control strategy to measure the process capability. Out of 3 stages, let's focus on the stage 2 of the process validation. This step includes 2 different aspects. The first one is the S-scale study. This one, we can run the larger scale operation at the manufacturing facility, usually in GMP area. We compare this data, process data, with the small scale data, as well as some of the pilot scale data. And also we compare some process parameter with some data from the process characterization. Second one is the site-specific and then the facility-dependent studies. This one includes a mixing validation, and then microbial, the mapping study, and the microbial control study. If it's a stainless steel bioreactors, we also testing the cleaning validation to ensure that all the product, all the residue from the stainless bioreactor is gone after the cleaning. Now, let's think about the connection between the process validation and then the process characterization. In the previous slide, I briefly mentioned that phase 1 includes the process development and then process characterization. Through the process characterization study, we can understand the relationship between the process parameters and the quality attribute during the bioprocess. The key activity of this PC study is establish the integrated the control strategy in order to maintain the good process capability. By this PC study, we can have the better strategy how to control the process with the high process capability. Quality by design, the QbD is covered by the ICH guideline, ICH guideline number 8, the pharmaceutical development, ICH guideline number 5, Q9, the quality risk management, and then Q10, pharmaceutical, the quality systems. As the various ICH guidelines shows, quality by design is strategic approach employed in the various industries, including the biopharmaceutical, as well as manufacturing, and then product development. The main purpose of this one is to ensure the consistent delivery of the high-quality product to the customers. This one involves the systematic and the proactive processing of integrating the quality consideration throughout the entire product life cycle, from the conception to the production. In the context of a biopharmaceutical, the quality by design focused on the optimization of the development and the manufacturing and the control process of drugs to ensure that these products are safe and then efficient and then deliver the high quality product to the patients. QbD is the QbD employs a risk-based approach to encourage the continuous improvement of the bioprocess and to design the capable, which is the robustness of the processes. The best way of the success for developing the good drug is to follow the robust risk-based approach, which is the QbD. For process development, since regulatory the body is recommending the sponsors to use the QbD approach from the process development stage. The goal of process development is the creation of the good process control strategy. Defining the QTTP, and quality target product profile, we need to find out the provisional or the CQA as well as the CPP, the critical process parameters. Risk assessment such as FMEA, we can find the initial CQA and the CPP. We can assess these parameters for the future process development. In the common technical document for regulatory filing, the process development and process validation, including the PC study, data will be the very critical portion of Module 3. For your reference, Module 3 includes quality information, including the pharmaceutical, the property, the manufacturing information, and stability data. During the PC study, and key and the critical quality attributes are tested as the main responses for the DO experiment. From the various experiments, the proven acceptable range and operating range will be determined. We call it the design space, including the operating range and the proven acceptable range. Oftentimes, researchers evaluate the process parameters and try to find out the edge of the failure to determine the boundary conditions using the DOE. These ranges are very important for impact assessment on the product quality, especially when there is the parameters excursion during the S-scale manufacturing. With these information, we can justify some of the excursion and also to provide some rationale for impact assessment during the deviations. Risk assessment is a big part of the QbD. The completing risk assessment such as FMEA that requires interdisciplinary team efforts. In practice, the main SME of the process share the process map before kind of internal meeting. Initial risk assessment will be reviewed by the interdisciplinary members, including QC and manufacturing and other related parties. They will brainstorm and they will provide some feedback for the accurate risk assessment. After risk assessment, the parameters for the PC studies will be determined for the more detailed PC study. In parallel with this risk assessment, the mitigation plan can be implemented to the process in order to minimize the operational failure, which to maintain the desired product quality. By the risk assessment, we have to determine which parameter will be tested for the PC studies. This is a risk-based decision made with considering the rating from severity and then control capability assessment. Severity is evaluated based on the product quality severity rating and the process performance severity rating. If the severity rating is high, we need to do the PC study for these parameters. But if the severity is medium, the PC study is depending on the control capability. If the capability is well controlled, we do not have to do PC study. But if the control capability is poor, we should do the PC study. Control capability is evaluated based on the likelihood of excursion occurrence as well as detectability and considering the manufacturing capability. The slide shows an example of the risk assessment and how we determine the parameters required for the PC study. This one is the risk assessment for Pro-A step, which is coming from the purification of monoclonal antibody. For the elution step, the pH buffer and pH conductivity and the peak start collection, the optical density was evaluated through the risk assessment procedures. Buffer pH is evaluated as a risk high since the severity of this event is high. That means this pH buffer pH needs to be tested for the PC study for the Pro-A operation. I explained that the main outcome of the PC study is to provide the scientific rationale for establishing the good PCS, which is a process control strategy document. This is a key document for the S-scale, the bioprocess during the tech transfer. key components of PCS is criticality and the range of process input and output parameters, media buffer prep information and equipment information, and a sampling plan. Many of the PCS elements are developed via the risk assessment. The PCS is a final outcome of process development as well as the PC study. As mentioned earlier in the previous the slide, this process control strategy is not the point-in-time activity. That means it's going to be the evolved over process knowledge accumulation. Once we are done with the PC study as well as the process validation, we'll get the more information on the process. This will make this PCS more robust, and then we'll provide the better information for process control the later on. The purpose of design of experiment is particularly understand the interaction between the two or the more variables, process variables. We use the different independent variable to understand the impact on the response variables. Usually, this DOE testing is used for the characterizing the process as well as optimizing the process. So this PC study, we also employs the DOE testing to understand the effect of process input variable on the output parameters, output responses. The designing an experiment involves 4 steps. The first one is select the study design, and the second one is set up the experiment. The third one is run the experiment. After completion of the experiment, we need to collect all the data and analyze using the statistical software. During this process, we need to consider the couple of things. The first one is you need to consider the independent variables and how these are related. Based on the hypothesis, we designed some experiment to understand the relation between the independent variable and the dependent variable. Also, we need to understand how to measure this dependent variable. So we need to have some good method to measure, monitor this responses. So in order to get the proper response, we need to have a method tech transfer from the client in order to the method development, analytical method development. There are many ways of performing the experiment for the PC study. Typical way of a study design for the PC study is OFAT, one factor at a time, the fractional factorial, and the full factorial design. There are pros and cons of each experiment design. These days, some of the statistical software like JUMP also the guide SMEs for the designing the experiment. For PC study, any design can be utilized for the study purposes, but this one is based on the risk assessment, which the parameters will be used for the PC study. If there's no interaction between the parameters, so we can use the OFAC. But if you think of some interaction between the parameters, we need to employ factorial design or the fractional factorial design to understand the interaction between the parameters. This is a typical the process characterization, the study flow. First, we have to set up the scale down model development as well as qualification. once we qualify the scaled-down model using the PC plan, we do the several experiments to understand the relationship between the parameters. In this study, we have an example of upstream. We can use the Ember 250 or the benchtop small-scale bioreactor as a scaled-down model. We can qualify this scaled-down model using the S-scale process data as well as the small-scale data. Once we qualify the scaled-down model, we set up the PC, the master plan. Based on the PC master plan, we'll run the many experiments. For the upstream process, we can do the several sets of PC study. The first could be the screening the PC using the screening DOE. Once we sort out some of the major parameters, we can do the RSM DOE. And then also we can use the OFAC to introduce some different parameters. For example, for upstream side, the feed amount can be the one good candidate for the OFAC study. Once we are done with the PC study, all the data will be collected and then put into the report form. This PC study report will be the source document for the PCS process control strategy for the upscale, the scale up. In the last session of my presentation, I'm going to show you the case study. This process shows a typical monoclonal antibody production using the continuous centrifuge followed by the depth filter and the startup filter with the downstream process with one capture and then two polish steps. I'm going to use the Pro-A chromatography as the case study for the following the case study. Okay, so some details of a Pro-A step in the DSP downstream process. We used the map select Sure LX from the Cytiva as a Pro-A resin, and then we used the HCCF from the upstream as a load material. As an outcome parameter, we used the process yield or the in-process testing. We tested the protein concentration, HCP, HCD, and the residual Pro-A concentration, and also some ICIF and the CE-SDS and the SAC as in-process testing. As you can see here in the table, we use the bed height with 20 centimeter. We reduced the diameter for the lab scale material. Symmetry is about the same. HETP is also the similar. The protein A step consists of equilibration, a load, and the wash 1 and wash 2, wash 3 step, followed by the elution. And then we're going to do the resin regeneration for the next cycle. I did not implement all the data, but out of all the analysis risk assessment, we found that 7 parameters here has a very high severity. So I selected the 7 parameters for the PC study for the next step. This one shows how we qualify the scaled-down model. We typically use the TOAST two one-sided testing for this equivalence testing. We use the larger scale data run and the small scale data run. And then the difference between the average of large scale data and then the small scale data. needs to be within the tolerance limit. In this figure, the light blue one is within the equivalence range, but other one, the yellow and the orange, is not equivalent process. Using this testing, we qualify the small scale down model as a qualified model for the further PC study. From previous risk assessment, we selected 7 parameters as a main independent variable, which is our load ratio, and then the loading, the linear velocity, the wash-it 2 buffer pH, and the wash-it 3 buffer pH, elution buffer pH, lastly, the elution linear velocity. Using these parameters, we did some of the DOE testing, and then here the output is HCD, host cell DNA. Using this DOE testing, we found that the load ratio and the wash to buffer volume and the elution buffer pH and the elution buffer linear velocities are statistically significant impact on the host cell DNA. And we also found that there's an interaction between the wash-to buffer volume and the elution buffer pH. This slide demonstrates that how we determine the criticality and the cross range after our PC study. On the left side, we have load ratio as a key parameters with AR acceptable range on the left side. After PC study, within the range of 30 to 55 load ratio, we found that this parameters load ratio is a very critical parameters to impact the KPI, such as process yield, as well as the host cell DNA. We define this load ratio, upgrade from the key parameter to the critical process parameter. In terms of the range finding, we used the proven acceptable range as 30 to 50 because the 30 was the low range of a PC study, and the 50 was the high range over the previous acceptable range. So we picked up a 30 to 50 as a proven acceptable range after PC study. Based on our historical operating range, we set our target range as 40 to 50 based on the historical data. So with these activities, we put some load ratio as a critical parameter with the target range of 40 to 50, with the proven acceptable range is 30 to 50. So these PC study data will be the good source document for the PCS in the scale-up study. Samsung Biologics is providing the PC service. Samsung Biologics has an MSAT lab which can provide the PC service for the client using the state-of-the-art facility. We have Ember 250, 4 Ember 250 unit, also the Actor unit, advanced unit for the downstream study. So the upstream and downstream, the PD lab has a good facility, as well as the SME, who has a very extensive experience in the PC study. As an upstream the service, we can provide some PC study, as well as some of the LIVUCA and then EPCB, extended end of production, the cell bank. And then we also provide some the base feed media stability testing, and then cell bank the compatibility testing. For downstream side, we can provide some the basic PC study through the risk assessment and scaled down model qualification. Using the DOE and OFAT, we can show some the relationship between the process parameter with the process output. We can run the confirmation run at the worst case study. Along with these typical the PC study, we can provide the small scale the resin lifetime study and the membrane lifetime study. And also we can provide the impurity clearance study, as well as VC. We also provide the virus clearance study the support. In case we need to do some reprocessing due to the FIT failure, we can run this small scale the reprocessing study from the MSAT lab. Get some conclusion from my talk today. The quality by design is a regulatory initiative for pharmaceutical development and the process characterization based on the risk-based assessment to control the process performance. The principle of the pharmaceutical development relevant to the QbD is described in many the ICH guidelines through the ICH Q8 to the 11. The PC study is establishment of criticality and then operation range for the process parameter in the PCS and the process control strategy. Generation is optimal and the PCS is essential for the successful process scale up. With this scale up, the proper scale up, the process validation, and then the commercial manufacturing will be pretty robust. With using this is the good PCS strategy. Samsung Biologics is a good facility. Has a good facility and a good talent for the PC study. The Samsung Biologics can be a good partner for the PC study as the service provider for the potential client, along with the larger scale manufacturing. Thank you for your attention.
All right, perfect. Thank you so much, Young-sun, for the presentation. A lot of really great information there. So we do have time to answer some of your questions now. So if you do have a question for Young-sun and weren't able to get it in during the talk, now is the time you want to go ahead and do so by using the box on your screen there. Okay, so this attendee, Young-sun, would like to know, says the QbD concepts may not be familiar to everyone. What could be a misconception of QbD that people have in mind?
As mentioned earlier, the QbD is the strategic approach that aims to ensure the high quality of product by employing the statistical and then approach as well as the risk assessment methodology. There are a few misconceptions that people have. The first one is probably the QbD is too complicated, because QbD is pretty much new to someone. The QbD is a systematic approach that uses science and risk management to gain the knowledge of the product and the understanding of the process. Once we understand this principle as well as the methodology of the QbD, we can apply it to many different areas. Not only for the pharmaceutical, you can apply it to the medical device, as well as some of the environmental monitoring for the manufacturing of the drugs. That's the first misconception. The second one is the QbD is only for the company. The small company, the SME, doesn't want to use the QbD because they think it's too complicated. But any company can use this QbD principle, and they can get the benefit from the risk management and then the continuous improvement of the process to produce the high quality product. If you do not have the good facility for the PC study, CDMO like Samsung Biologics can provide some customized service for the process characterization for the company. The third one is the QbD is only for technical professional, like the scientists and engineers. Managing the quality of the pharmaceutical is not only for the scientists and engineers. This is something that everyone in the organization should be get involved to make it happen in terms of like a product quality management. The key is the mindset of the people who are engaged in the product development as well as manufacturing of the drugs at the larger scale. So implementation of this mitigation plan to lower the risk for the pharmaceutical manufacturing needs cooperation of everyone in the company. I think that's the key three major misconceptions that people might have.
Okay, great. Thank you so much for that answer. Moving along here to the next question. When process characterization studies are performed, you say you run in many experiments to find out the design space of each unit operation. So how do you need to select DOE or OFAT for PC studies?
During the presentation, I explained that there are several ways of doing the PC study. One would be the design of experiment to involve the process parameters all together at the one experiment. The other one is one factor at a time. So the one process parameter is only used for the testing. There are the few advantages of DOE testing over the OFAT, the one factor at a time. The first one is obviously you can reduce the number of experiments doing this DOE testing rather than the OFAT. The other one is by using the DOE testing, you can understand the interaction between the parameters. There are so many parameters to determine the quality of product. So you need to understand the interaction of these parameters to how to impact our final quality. So that's going to be the second advantage. The third one is you can predict the process performance. So by doing the DOE testing, we can get some help to predict how and how these parameters can predict to the process. That can be identified with this DOE testing. The last one is probably you can derive some statistical model by doing the DOE testing. That's another advantage of DOE testing over OFAT. But how to use the OFAT over DOE should be coming from the risk assessment before the PC study. And the OFAT approach is also that can be used when when we tested the parameters which is not have any interaction with other process parameters. For example, the bed height of the chromatography can be used as OFAT testing. Also, the feed amount for the production bioreactor in upstream can be also tested with OFAT. But there is not a golden rule to which one is better. In practice, when you do some study with the client, we need to discuss with the client how to set up the experiment, either DOE or the OFAT, based on the risk assessment as well as the process understanding.
Okay, great. Thank you so much for that answer, Young-sun. Moving along here, during your talk, you also emphasized the importance of process control strategy for scale-up of bioprocesses. And these days, PAT is being used for process control of bioprocesses at scale. And I'm wondering if Samsung Biologics is also implementing PAT for manufacturing its biologics.
The short answer is yes. As you know, the PAT, the process analytical technology has been defined as a mechanism to design, analyze, and control the biopharmaceutical manufacturing process through the measurement of a CPP, the critical process parameter, which affects critical quality attribute. This PAT is aimed at understanding a process by defining their CPPs. Accordingly, the monitoring them in the timely manner, preferably the online or the inline. This is going to be very efficient in terms of monitoring the parameter as well as controlling parameter during the process. At Samsung Biologics, we also implemented some PAT technology. For example, the Raman spectroscopy to monitor the metabolite, usually the glucose and the lactate, and then biocapacitance probe to measure the cell density, and then the PCO2 probe, which we can measure PCO2 concentration in the production bioreactor. Some client wants to use these PAT for just monitoring purposes. Some clients want to use these PAT technology for the feedback control. Actually, we as the CMO, we have the client who use it either way. So based on this, the client's process, this PAT tool can be used for like I said, the monitoring purposes or the controlling purposes. Like I said, the Samsung Biologics has a capability to implement this PAT technology based on the client's needs and demands.
Okay, great. Thank you. So we have time for one more question here before we wrap things up. This attendee says that as part of PC studies, you mentioned qualification of the scale-down model is critical to the study success, and would like to know, do we need to qualify the scale-down model for each drug product?
As mentioned earlier, scale-down model is a small system that represents the specific operation step in a process without using the large amount of the materials, especially for the downstream. In theory, the scale-down model should be qualified specifically for each product, but if the customer is using the platform technology for its process development, scale-down model for the PC study for the first molecule can be leveraged for the following the product, which has the same platform. In theory, that's what I'm thinking, but you may need to perform some bridging study to demonstrate the previous qualifying the scale-down model works for another molecule if it's necessary.